Thank you. Can you all hear me? Yes? I'll move that up a bit. Thank you very much for the invitation. Thank you for coming out on a rainy night. I'm hopefully going to take you away from this weather and take you to the South Pacific where I do my work, where there's certainly a lot more sunshine than there has been. So I'm actually not going to start in the Pacific. I'm going to start in the Indian Ocean. So this drawing is from 1598, when Dutch sailors first set up their outposts on the island of Mauritius. And you can see they're having a rather jolly good time of it. It's the pictures are called parrot hunting on Mauritius. So this is the Mauritius grey parrot, and you can see people running around with clubs, clubbing them out of the air. They're riding a giant saddleback tortoise, or sitting in the shell of the tortoise. And sorry, I should have turned this on. There we are. And in the background here, you can see these funny-looking birds represented that look something more like a cassowary, but actually is meant to represent a dodo. By the middle of the 1700s, all of these organisms were extinct. And, you know, the stories about what happened on Mauritius are, well, incredibly sad, but it also encapsulates this feature of island forms called the island syndrome, which I want to talk about. Part of the reason, not the entire reason, that these organisms succumbed so quickly was that they were incredibly easy to catch and kill in great number. They were very vulnerable to introduced predators that humans brought in with them as well. If we think about the dodo and what happened there, the dodo is, this painting here is actually one that was one of the few that were made when the dodo was still extant, so still alive in Mauritius. But it was drawn from a bird that was in the menagerie of Emperor Rudolf II in Prague, and it's the Jakob Hofnagel dodo. So hopefully you all know something about the dodo, but this endemic bird from the island of Mauritius, where I just showed you the picture from, and it went extinct very quickly in the few decades after the Dutch arrived, and the last confirmed sighting was in 1662. So it's quite stunning that, you know, this bird met its demise so quickly, like many of the other species there, and we could think it's just a really sad case. It's like this, you know, evolutionary novelty. Sad that it's gone for good, but you know that this kind of thing happens. And when we think about the dodo, we might think of it as something like just really bizarre that only occurred in this one place, and the dodo certainly did. But if we look at, you know, where did the dodo come from? And it turns out that it's a pigeon, in case anyone didn't know that it was a pigeon, and the closest living relative of the dodo is something called the Nicobar pigeon, which is over here in Indonesia. So what we think, and they're separated by about 42 million years. So what they think happened is that a flighted pigeon ancestor made the journey over here to Mauritius about 8 million years ago, not long after the island was first formed, and hit play on the button of evolution, and you come up with this dodo. So far from being an only child of this kind of evolutionary trajectory of what happens when you put a pigeon on an island, on the neighbouring island of Rodrigues is the Rodrigues Island solitaire. So this is the cousin of the dodo, and it also emerged from that colonization by a flighted ancestor 8 million years ago. Also flightless, also extinct now. And so the dodo, while unique, had a sister species on the neighboring island. Now, if we move across into the Pacific, what they've found is that there are extinct, flightless subfossil bones of a giant species of duckula, aves, a pigeon in Fiji. And in Samoa, there's something called the tooth-billed pigeon, or sometimes called the Samoan dodo. This one is still extant. It is, not in a good way environmentally. Its populations are declining, and so it's nearly extinct. And it's also not quite flightless, but nearly flightless. It is able to fly, but mostly forages on the ground. But what this tells you, apart from a conservation story about flightlessness on islands or becoming near flightlessness, is that the dodo, while unique, is an example of what repeatedly happens when you put a pigeon on an isolated island and leave it to evolve for some time. And that kind of evolutionary journey of these repeated patterns of evolution is something I'm going to talk to you about tonight, not in these pigeons, but in other birds in the South Pacific. And it happens actually in lots of different birds across the taxonomy of birds across the globe, but also some of the patterns that I'm going to talk about are equally frequent in things like mammals and sometimes reptiles as well. So for this talk, what I want to do is introduce some concepts about evolution of island-dwelling species. I'm not sure how many biologists are here tonight. I know it's a broad scientific society, so hopefully I can go into enough detail, but not bore the biologists. So the two things that I want to talk about are something called taxon cycles, the idea that species themselves go through these defined stages that can end up with just an extinction event, and then another one comes in. So the species go through these ages, which I'll explain. And then the island syndrome, which I've alluded to, this suite of changes that occur, that commonly occur, at least when birds, mammals, other species are on islands for an extended period of time. And then I want to introduce the main group that I work on, the white eyes, but a specific species of white eye called the Silvereye, and how I'm using modern genomic data to test some of these kind of old ideas in kind of island biogeography, taxon cycles, and island syndrome. And then just at the end, I just want to spend a couple of minutes reflecting on the fate of island species. So first of all, to think about the taxon cycle and to explain what it is. So the taxon cycle was a concept that was introduced by people like E.O. Wilson and expanded on by Jared Diamond. And what they talked about was that there are these phases that populations go through, and the first phase are called supertramps and tramps, and these types of species have really high dispersal ability, so they get to a broad geographic area, but they tend to have low competitive ability. So these represent islands up here in the top right, my cartoons of islands, and then where the distribution of these tramps and supertramps is coloured green. So they'll get to like the peripheral islands and in the kind of edges of larger islands once they become a tramp. This would be the tramp stage, this would be the supertramp stage, but they can't kind of penetrate they don't have the competitive ability to kind of penetrate forested areas or specialize at high altitudes and things like that. And so their geographic range is increasing, but they have such high dispersal capacity that their levels of population differentiation, which is what this kind of cartoon is meant to represent, is actually very low. So there's lots and lots of gene flow connecting the population, so they don't have strong population structure. It's thought then that some groups will go into what's called the great speciators. And this great speciator stage is where you start to get some divergence, so the different colors of green, so population level divergence is increasing. But the geographic range is also kind of maximal. So they've had very good dispersal capacity still, but dispersal then, once they get to all these places, dispersal starts to drop off and they start to diverge. Sometimes they can go through cycles of these. They can expand again and go back into the super tramp stage and go round and round. But others go through this specialization stage where they speciate. They might become, to the internal parts of the island, and then a relictual stage where they might be confined to mountaintops on the islands, and then their populations or their species are very vulnerable, and that's when we see these kind of single island endemics restricted to maybe only a small part of the island, and eventually they're probably going to go extinct. So through this process, we have this rapid, I should have made this even narrower, but this rapid decrease in dispersal ability. So while they're down here, they can't really disperse very well. So the great speciator stage, though, is a really interesting stage in this taxon cycle process. And so this idea, even though it says great speciators, it's actually great subspeciators. So it's one species that has many, many subspecies. And in their book on birds of northern Melanesia, Ernst Mayr and Jared Diamond did these analyses of bird distributions. This is golden whistler, pachycephala, and you can see there's a lot of morphological variation that has been described as different morphological subspecies, but the species as a whole has this really wide geographic distribution in northern Melanesia. And they described this stage of the taxon cycle as a paradox. And the paradox is that how can you have a wide distribution that implies high dispersal ability, but lots and lots of morphological variation and subspecies that implies that there's been lots of divergence happening, and most models of divergence and speciation would require some reduction in gene flow and dispersal to start off with. So, the obvious solution to this paradox is that you have some rapid post-colonization change in dispersal ability. And that it might just be a behavioral reluctance to disperse, but over time they might be physiologically and morphologically unable to do so. So in an extreme case, you end up being a flightless bird, for instance, or a flightless insect. I mean, there's many cases of this kind of extreme reductions in dispersal capacity and propensity. And what might underlie that? Initially, it might just be a behavioral, like I said, but it could be due to genes or plasticity, epigenetics, development, not a field that I work in. But the idea that it happens so quickly sometimes suggests that it must be acting on kind of standing genetic variation, things switching on and switching off eventually. So that's one idea, and it's related to this next idea that I'm talking to you. So the idea of taxon cycles, great speciators, and specifically changes in dispersal capacity, which is something I'm very interested in. So this leads me into another kind of concept in thinking about evolution on islands, which is the island syndrome. So the repeated reduction in dispersal that in island birds is just one component of the island syndrome, but it actually encompasses a range of ecological changes. So we think about species broadening their ecological niches. This is a picture of one of Darwin's finches, the vampire finch. So what it does, it plucks out the feathers of the back of these seabirds and drinks their blood. So that's an extreme change in ecology, but adds to the wide niche of Darwin's finches as a whole. Reduced dispersal can be considered part of the kind of ecology as well. we have something called the island rule, and this is probably the component of the syndrome as a whole that's been studied the most in the most taxa, and it seems to be robust certainly in birds and mammals, and lizards actually. So the island rule is that the expected body size change will be, if you start small, you evolve to larger body size, but if you start large, you evolve to smaller body size. And we can end up with kind of giant examples of something on an island, or miniature examples. So in the Mediterranean islands, there were tiny little elephants and deer and hippopotami. They're all extinct now, sadly. So this is a widespread, it's called the island rule, started out being called Foster's rule when it was described for mammals. And these are some of the birds I'm going to talk about. These are both Silvereyes, but two different subspecies. This is the island one, which is only 80 kilometres off the coast, and I'll show you where these live off Australia. And the mainland one, we have the opportunity to get these pictures occasionally because it's only 80 kilometres away, and vagrant mainland ones arrive. And there's about a 40% size difference in these things that are still considered the same species. Other morphological changes happen in birds in particular. We don't have the big differences in the plumage dimorphism that we might see on mainland area. We see more rounded wings rather than pointed wings, and this relates to dispersal capacity. If you have long pointy wings, you're more able to go longer distances. And something that's called a shift to this graviportal body plan rather than the opposite would be cursorial. So kind of it's all about the relative sizes of different skeletal measurements. So when I try to describe graviportal, it's like I describe myself as graviportal, kind of a bit shorter and like shorter limbs and everything. So that seems to happen. The relative size of different body traits changes. We also have big changes in natural history, life history, and by that I mean all the stages throughout the life that are things like how quickly you grow, when you might fledge, when you might leave your parents, what age you are at first breeding, what the average lifespan is for an organism. And what seems to happen on islands is that there is a slowing down of life history. Instead of breeding young and getting out as many offspring as you can and dying young yourself, you put more into your own survival and then more into each of the fewer offspring that you produce. And this can also result in a slower metabolic rate, although empirical data on that we need a little bit more. And then finally, the behavioural things that I've alluded to, like with the organisms on Mauritius, this apparent tameness. It's really predator naivety. So without predators in the system, which many islands don't have, or at least traditionally didn't have predators, then there's no need to maintain predator responses, vigilance, and you end up, these are my hiking boots, just sitting in the forest, and this is a Lord Howe Island woodhen that will just walk up to you. Unsurprisingly, they brought these things back from the brink of extinction because, when people came there, this is a nice chicken-sized meal that walks up to you. So so they have these changes in behavior as well. So how do I test these components of these ideas? They're very intuitively attractive ideas about how evolution will occur and the patterns that we expect to see. But what I wanted to be able to do was say, well, okay, in taxon cycles, the solution to the paradox is to invoke a change in dispersal capacity. Can we find a genetic basis for that would help us understand rapid switching? And then in terms of the island syndrome, of which dispersal change is part of, what mechanisms allow for that repeated change at a genomic level? And there's one kind of slight diversion I want to go on to think about the importance of stochasticity. Because when you think about repeated patterns happening over and over, you probably want the right genetic variation there for that to be able to occur. But when species colonize islands, they might be subject to strong drift during founder events themselves, or as remaining small populations over time. So can we find a system where we can actually test what the genomic and impacts of founding are on genetic variation? And so when we're thinking about the repeated changes, one way that it could happen is reshuffling of standing genetic variation. So there's all this genetic variation in the source populations, and that these newly colonized populations take that variation and reshuffle it in different ways to get similar phenotypes. And that's a faster way for evolution to happen than to stand around waiting for a novel mutation to occur. However, novel mutations may also contribute to some patterns of repeated variation. Okay, so the system that we'll go now to, well, first of all, you may not all be familiar with this family of birds, the Zosteropidae. This is their distribution, both the grey and the orange. The orange is just where the Silvereyes are, so this is where I work. But it's a really, really large distribution, and they have radiated in about most of the species, of the 100 or so species in the group, have radiated in less than 2 million years. So this is a really, really fast radiation. And in fact, it's, apart from cichlids, which are the fastest radiation for a vertebrate, this is number 2. And they're very good at dispersing over wide areas, and they're really, really good. You can't quite see, but on most of these little islands, there'll be some white-eye form all throughout here as well, and all throughout here. And what that means is we end up with lots of cases where we have, in this case, a Silvereye and another white-eye on the same island. On this little island, we've got 3, a big one, the Silvereye in the middle, and a little one. So white-eyes in general are just really good at colonizing and dividing up the environment and kind of dominating, really. And here's another species pair from Vanuatu. So the Silvereye in particular, why it's useful for answering the questions that I've talked about, or addressing some of these bigger, older ideas, is that it's kind of done a repeated experiment for us, as much as you can in a natural system. And it has colonized islands of the Southwest Pacific over a spectrum of times. And the oldest ones are out here in New Caledonia and Vanuatu. So together, these are southern Melanesia, and Mare and Diamond worked up here in northern Melanesia. There are some really old subspecies on, like Lord Howe Island, Heron Island. That was the one that I showed you with the 40% body size increase. And then we can easily sample the mainland. And there's a different subspecies on Tasmania, which very conveniently, in the 1830s, started doing a sequential and historically documented colonization sequence of all of these places. So sometime in the 1830s, it's a migratory subspecies, so it should be flying this way. Sometime in the 1830s, a flock went this way and established itself very quickly on New Zealand, and then spread out, getting to Norfolk Island in 1904. So it's really useful for looking at both the great speciator concept and the island syndrome concept, which I'll show you in a minute. But back in my Supertramp days, I had to island hop after all of these birds, and so got to spend a lot of time out in the field, and it was all great. These are mist nets. This is how we caught the birds. This is how I traveled around island to island, or walking, or in a boat. There were lots of things to be careful of. This is not a cloud. This is a volcanic plume. So lots of volcanic action, always trying to work your way around and stay relatively safe. But the kinds of, the geographic extent that I went on, I mean, I went to lots of these places, but I just, want to say I could not have done it without a huge amount of help from local communities and people that came along, and different students and collaborators along the way, and just really benefited from the local knowledge, which people were very, very happy to share. Okay, so in terms of the taxon cycle, what is the Silvereye. So I've alluded to the fact that it might be a great speciator. So why do I say that? Well, one, it's got a really broad distribution. All these different colours represent different subspecies, and you can't really see there's some on Lord Howe and Heron Island, and then a few in Vanuatu, New Caledonia, one out on Fiji. I never got to Fiji. It's the one location I haven't got to. They kept on having a coup every time I tried to go there, a soft coup. I tried to convince my employers it was worth it, but they always said no. So it has got this broad distribution and lots of subspecies, lots of morphological subspecies, which falls into that great speciator concept. However, so somewhere here over on great speciators. However, this colonization in the last couple of 100 years of New Zealand and outlying islands is like one of these expanded phases. So what that portion of the distribution has done is kind of gone back into this super tramp phase. So, you can do cycles of this, whereas a few of them are on their way to speciation. In fact, some people argue that the one on Lord Howe Island should be elevated to species, and perhaps the one on Heron Island as well. So we have a group that's kind of covering a span of this taxon cycle, but would primarily be called great speciators. So the question was, can we somehow document this genetically, any change in dispersal? And is it kind of specific to islands? So one of the first things we did was just to look at the population genetic structure among islands and across the continent. So birds sampled all across the continent of Australia. And FST is a measure of genomic divergence. So if it's down near zero, there's free gene flow. If it's up higher, then there's restricted gene flow causing structure among populations. And what we see over kind of similar timeframes is that the island ones water barriers generate a great deal of population genetic structure and elevate that amount of genetic divergence between them. So water barriers are really effective at stopping or minimizing or causing dispersal to decline. And it might be that they just, they got there quite randomly or luckily, and they never really like to cross water barriers. But why we think water barriers aren't an inherent problem for the species is that I mentioned there's this migration from Tasmania. So this group crosses the Bass Strait every year and comes back. So that's not a problem for them. We have this regular arrival of mainland birds on these barrier reef islands out here, and we have the multiple recent by the last couple of 100 years long overwater colonization events. So we think they have really good overwater dispersal ability, but sometime after they get to the island, they just switch it off and they start to diverge, causing these patterns. And I should say that these are banding returns, so these guys can get around, like these are huge distances in Australia. So to first of all, to be able to say something about, what's happening genomically with dispersal, first of all, we need to figure out what the phylogeny looks like. And so this is just to point out that we really do have two groups. We have this kind of Australian, we call it New Zealand outlying island group, and we have this southern Melanesia group, and they are quite separate on a phylogenetic tree that we build. And so what we wanted to do then was so any differences that we find, so we know that these ones, like on Lord Howe Island, they don't disperse, for instance. We know that these ones don't disperse. We know that these ones are really good at dispersal. But any differences might be due to just the differences in the genetic groupings that we have here. So when we were looking at the genetic basis of dispersal, we started by looking at candidate genes, so genes that are known in other groups, humans even, that are associated with dispersal, migration, explorative behavior, Sometimes there's some personality ones like boldness, so that organisms will kind of get up and go. And to see if there was any relationship between the variation of these candidate genes and what we knew about dispersal in these populations. So what this one here is showing, there's this candidate gene called CREB, which is involved in circadian rhythms and things like that. These bars down the side represent population structure. So these are the two large groups. So we have this Southern Melanesian group here, and we have this Australian, New Zealand, outlying island group. And then what these dots here are showing is the relative lengths of this particular, it's a microsatellite gene. So they come in repeats, and these ones have more repeats and these ones have less. And so there's this whole group here, including two of these blue ones that tend to have short repeats, and they are not dispersers, and then these are the highly dispersive ones. So it seems like there's something in CREB1 that might be a switch, and it's not related entirely to population structure, because we've got these two that are in this group phylogenetically but are in the shorter CREB length. And we were also able to look at another candidate gene, this time not at a population level, but at an individual level. So the birds in Tasmania, they're partial migrants. So some of them migrate and some of them don't. And so they self-sort for us. So we can go in winter, and the ones that stay in Tasmania are the non-dispersers, and we can catch that phenotype up on the mainland, and they're the dispersers. And so what we found was that there was on average, I mean, there's a bit of overlap, but on average, the ones that tended to do these long migrations tended to have these longer allele lengths. So these are candidate genes where we're just kind of trying to find information from other forms, and so that's not ideal in that we would like to look across the whole genome. We're currently processing that now. Okay, so I mentioned founder events and drift, and the sequential colonization of Silvereyes allows us to follow what happened in that sequential colonization with respect to genetic diversity. And all I want to say really from this is that if you look on the right-hand side, we can see what happens. These arrows are like colonization events and how they go sequentially. And over time, over sequential colonization events, you do get a reduction in heterozygosity and a bit in allelic diversity, but it's really quite hard. You have to put them through sequential colonization events. And they don't lose much variation at any one step because they're colonizing in flocks and their populations are recovering very quickly afterwards. Perhaps one slight exception is this one, which is the only human-mediated introduction in the series. And just as an aside, this was done by a very interesting person, Eastham Guild, who introduced over 45 species, over nearly pushing 1,000 individuals of birds to Tahiti for no apparent reason. 14 of those took, of which the Silvereyes is one. And of course, these are not good birds to have there because they have a big impact on the endemics. Okay, so that's our first foray into the taxon cycle idea, and we can really, what we want to really get down to is what's the mechanism that's driving the or can we find, the proximate mechanism that's resulting in this reduction in dispersal? And we think we have hints, at least, that it could be a genetic switch. Thinking about the island syndrome, like I said, there's many components to that, but the one that we've focused on is this body size change, and that's shown in that picture there. But this is bill size at the bottom and body size summarized in the principal component, so a whole lot of measurements taken down and collapsed. And you can see the size of the mainland ones here, and as you go out to these islands, they're larger and larger. And these ones in pink are what's happened very, very recently. So they have a rapid evolutionary change in body size. So what we did here was to do whole genome samples. So this was 306 from across that range, where we did sequenced the whole genomes, and we had the morphology of each of those individuals as well, and we did what's called a genome-wide association. So looking to see if there are parts of the genome that are highly associated with large body size or large bill size. And essentially, you get this big each one of these is a SNP, a single nucleotide polymorphism. And as you get these ones emerging up here, especially if they get above this kind of significance line, these are regions of the genome that are associated with larger body size or larger bill size here. There's a high one there. This one I'm going to talk about a little bit more, this really, really highly significant one. And so what this is telling us, though, is the question was, when we're trying to find variants that are associated with the repeated evolution of large body size, are they coming about from standing genetic variation, or are they coming about via de novo mutation? And what we find is that a lot of these ones, they're shared across all the populations. This one's not. This is only in a small subset of populations, which I'll talk about in a minute. So most of the time, to achieve larger body size, they're using these variants that pop up in all the different populations, and they're just recycling them. So they're not waiting for new mutation. It's a really fast way to have a phenotypic change. And they're doing this in populations that have only been there for 100 years and ones for 100,000 years. So they're making the most of this genetic variation for large body size. So we went and we looked a little bit closer at this one really highly associated variant of this whole region here that's been dragged up. And we were able to drill down to the particular single nucleotide polymorphism and say, is it CC, CT, or TT, and what the body size was. And what we find that if you're a homozygote for TT, you tend to be larger. If you're heterozygote, somewhere in the middle, and there's quite a wide variation there. And they only pop up in Vanuatu over here. Part of the problem with doing these kind of genome-wide association tests is that you have to account for population structure. And we've got really, really strong population structure with these deeply divergent types. So we thought, well, this might be just because of population structure separating Vanuatu from everything else. So what we went and did was actually just went into Vanuatu and into part of it where there's a lot of gene flow, so population structure is not a problem, and we find the same pattern. So we're quite convinced that we've hit upon this novel mutation that only occurs in this archipelago that gives these groups an extra boost in terms of large body size. And so we wanted to then try to figure out if this mutation has a signature of what we call a hard selective sweep. So it's really favored. We think large body size is favored, and then once that mutation occurs, does it sweep through all the populations? And one of the things we can do is we can build like little mini phylogenies just for that region. And so this one is what the private region means for the novel mutation. And this is an example of an associated region that is shared among all the populations. And what we see is this funny little long branch here that indicates that it's only occurring in this one region and that it's evolved really, really fast. And we can also look at patterns of FST around this part of the genome. And I don't really have time to go into it now. I'm checking the time. But this kind of lumpy shape here is what the pattern that you would expect for a hard selective sweep, whereas this more U-shape, where it's called a peak-valley peak pattern, is what you expect when it's standing genetic variation and you have some variants that are segregating against lots of different genetic backgrounds across the populations. And we can then look at, drill down even further. That's the graph I just showed you, the high FST at this particular point. But it's got low genetic diversity. This is one of the tests we can do to GMSD, looking for negative values. And then there's a composite test that draws all these things together. And we can look at patterns of linkage disequilibrium. So this is where parts of the genome are inherited together just because they're physically close. And if something is under strong selection, it might drag the other parts of the close parts of the genome with it. And chromosome 2 is where we have what we think is the novel hard sweep, and it has really high linkage disequilibrium compared to the ones that are just sorting on standard genetic variation, which is lower. Okay, so two ways to use or access variation that's associated with large body size. Recycle what you've already got. That does seem to be the main way. And if you're lucky, like in Vanuatu, you might just get the right mutation that makes you bigger, even bigger still. There's one other way that might be important in these birds that we're just starting to look at. One other way to access some genetic variation that could be important in driving large body size or evolving large body size. So I mentioned all these cases of secondary sympatry, and in each of these cases you can see the Silvereye, and they're with either one or two other white eyes. Now some of these, like these ones here on Norfolk Island, you've got 2 million years divergence separating these forms. They are good species, but the Silvereye arrived in 1904, and when it arrived, there were actually 3 species it became the 3rd of the species of white-eye on the island. This one went extinct in the 20th century. This one's still alive, and then that's the Silvereye. And so that was 1904. In 1912, they did an expedition, and they collected these are in the American Museum of Natural History, and both plumage and some of the morphology, they decided they were intermediate and that they could be hybrids between these two. But in the paper it was from 1970, but looking through all this old material, they said hybridization between these two species resulted immediately after the Silvereye colonized, but has since diminished or ceased. It hasn't diminished or ceased. So we've been able to show that there is ongoing, this graph is showing across all the chromosomes lined up, everything above this dotted line is highly likely to be introgression, hybridization between the two, so shared between these two. So that's a story that we're following up on. We've now gone and got museum material, even from this one, to see if he was involved as well. He may well have been. So, that could be a case of ghost introgression. The species is gone, but his genetic material might still be there. Okay, so I'll just skip over this really, really quickly. I just want to take a couple of minutes to think about the fates of island species. But this system is being very useful for starting to pick apart some of the mechanisms of the taxon cycle and understanding why we should even get a repeated pattern, or how we get repeated patterns when islands vary so much, and importantly, how it happens at a genetic level. So I just want to take a couple of minutes just to reflect on the fate of island species. So since 1500, it's probably an underestimate, 128 bird extinctions on islands. 128 extinctions, 6 on continents. So some of you might recognize some of these species: Carolina parakeet, the passenger pigeon, and that one is a grebe from Guatemala. Islands have 122, and people have done various analyses of risk and how much more risky is it then to live on an island. It's really risky to live on an island. And part of that reason is the island syndrome. So have these organisms backed themselves into an evolutionary corner from which they can't escape when the goalposts move, when humans bring things to the islands. And this is the island of Guam in the South Pacific. And here are some examples of some of its extinct species, including a white-eye. And of the 12 forest bird species, so it's a small island, but 10 have been extirpated, and the last 2 are functionally extinct. Their populations are so small that they don't do what they used to do. And the culprit is the brown tree snake that was introduced during World War II when they were moving lots of equipment, when the US was moving lots of equipment around, probably Australians too, we won't blame the US for everything. And so now there's only 2 little vertebrates, little skinks left on the island. These things eat everything. The forests are silent and just dripping with spiders' webs, right? Because there's nothing to eat these insects. So in a situation like this, it's just gone too far. There's not much hope. But in other places, birds are responding in an evolutionary way to introduce predators. In this case, this is a lepeo on Hawaii, probably pronounced that wrong. And this is a heritable trait of where you nest. And in the last 16 years, since in this one particular site, rats were introduced, and they've seen it evolve higher nesting, so it might survive that predation. Also, another Hawaiian example of introduced disease, where malaria was sadly introduced. The mosquitoes were introduced, which was the vector for malaria. This bird, the amakihi, has high altitude populations and mid and low altitude populations. There's no malaria up here because the temperatures are too low for the malaria to complete its life cycle, plus the mosquitoes can't live up there. But this bird is, the ones down lower, it's one of the few that survived this mass extinction in the honeycreeper radiation. It has some tolerance now to the avian malaria. So the hope is that they can kind of more accurately characterize that and maybe do gene editing, who knows. And just, climate's changing, so the mosquitoes are moving up, so they need to do it soon. So coming full circle back to De Bruy's picture of parrot hunting in Mauritius. And I think it's really important to reflect on these times of overexploitation. These people maybe had an excuse. They didn't know. We still do it. But for me, I don't directly work in conservation biology, but I feel it's really important to understand how these things evolve, to understand the processes of evolution, to be able to unpick the mechanisms at play, to really be able to, potentially protect some of these species. It's interesting in and of itself, but it may well protect them. And the other important thing to remember is that, a lot of these species were portrayed as really stupid, dumb, just waiting for, they're asking for it, waiting for extinction. They were really ideally adapted to the situation and the environment that they had before humans altered it. So they're not some kind of dregs of evolution. So these kind of really interesting island forms are something that we should try to protect and understand how they evolved. And I particularly want to thank 2 former PhD students of mine who contributed a lot to the genomic work, and say thank you for your attention. I'm happy to take any questions.